Well, I want to begin by just acknowledging what a fabulous conference this is and how honored I am to be a part of it, and in general to have been a part of the, participated in the CFAR group. It's really been a very fascinating, stimulating intellectual experience, and I think we're at a very interesting intellectual moment in the field right now. I think this conference is really doing a lot to maybe clarify some of that. Let's see. In fact, I'm going to start by three quotes that I heard yesterday. Three different fascinating observations that I think sort of bracket my talk, and then I will work off. One was Tom Boyce's beautiful presentation yesterday, and talking to him afterwards. This concept of that hierarchies may well be fixed. We have dominance and subordinates, but the degree to which those hierarchies are virulent, if you will, have negative or possibly positive effects, that may vary, and that's a very interesting thing, I think, to think about. Where's the plasticity in our social organization? Last night, Bruce made something like this. I think I'm paraphrasing him. At least the brain is even more plastic than we know, and I think I'll hopefully give you some evidence today to show how that is so, and how it's still unfolding just how plastic, how complex and dynamic different kinds of activities are in our brains. And then I was touched by hearing these this expression from Fraser, apparently in his last days, that it's got to be the microRNAs. Well, that was kind of gave me chills. I'm going to end my talk talking about microRNAs, and so that's I don't know, an interesting little stimulus there. So I do work on songbirds, and I'm going to jump right in and show you a video of their social life as we study it in the lab. Let's see, is there a pointer? There we go. Yeah, okay, so some basic key points to observe are that these are very social animals. This is a male of the orange cheek patch. Here are a couple of females. We'll see what happens here whenever we put them, whenever we just look at what they're doing in the lab. We house them in groups of 20 to 100. It's kind of noisy. They're very active birds. See two males here. Here's a female. Yeah. He's going to kind of sing in a minute. He's working. There's a female. He's interested in that female, right? There's a song. Okay. She's kind of reacting there. He's kind of, get out of the way, bud. Yeah. Okay. Yep, she's back. So that would be directed song. He's directing the song clearly to this female. Here they are. This little allopreening, this female grooming this little guy here. And all this is going on is this cacophony, right? This fabulous social life. And now you might wonder if this is purely an artifact of housing them too densely in a lab, but it's not so at all, because if you look at what they do in the wild, this is what they do. They're native to Australia, the outback, and they live in colonies in the wild. And these colonies migrate around, often in search of water. They migrate around Australia, but they have this very again, the very close-knit little communities, and in those communities now, they actually form lifelong monogamous pairs. So there's this social richness going on with this pairing also occurring. It's kind of like humans, right, in a lot of ways. And this is where this concept of hierarchy has really fascinated me. And it continues to in the context of hearing Russ's wonderful work with the fishes, and we hear about the social hierarchy. It isn't at all obvious that these guys have social hierarchies. People have looked for it. You know, I'm not going to talk about a study that we've done, haven't published yet, but trying to just get an ethological description of what the guys in the lab are doing, these guys. But we don't see a lot of hierarchy, yet they obviously have social structure. And so this is really kind of a side issue. I just had to brought it out today, given the talk yesterday. The main reason, of course, that we study zebra finches and other songs is because they're songbirds and they communicate through this vocal behavior. Communicating through vocal learning is a relatively restricted ability in the animal kingdom. We do it, and a few other species do it, but no other species does it that's as amenable to study and, as the songbird. And the zebra finch has become the main laboratory songbird. In the case of the zebra finch, here's the male up here, and here's a female. Now, only the male actually sings. Both sexes vocalize in different kinds of sounds, but only the male does this particular thing called song learning. And just to give you, and this, I'll also say that every male ends up singing a slightly different song. So each male's song becomes his own kind of unique identifier. Zebra finch, song 1. Zebra finch, song 2. Well, I think it's obvious why we call it song, right? It's really quite melodious, but something only a mother could love, I guess. I don't know. It does kind of grow on you. I don't know. But so every male, though, sings a different song. Now, he learns that song during a critical period. We heard a beautiful story about critical period work yesterday from Takao. There's a deep story about critical period learning in the context of the songbirds. The young zebra finch copies an adult tutor, usually his father, during this relatively narrow window of about a month when he's an adolescent. If he were, you know, a human, he'd be learning to drive a car and running into things. And in this case, he's learning to try to copy the sound that his dad is making. It's never a perfect copy, and this is important. So he winds up singing, you know, his own variant of that. But this isn't the only kind of learning and the only way that the song communication is used, because after all, these guys continue to sing throughout their lifetime, and both males and females will discriminate their mate from their neighbor, from an unfamiliar bird, somebody who's new to the colony, somebody who's been around a while. And we've been able to show this behaviorally and at a molecular level as well. So there's a constant process of using these kinds of auditory, sensory, social signals to mediate different kinds of social interactions. So I'm going to talk today, I'll give you 3 little vignettes about the illustrations of the role, at least, the potential role of epigenetic processes in being driven by and impinging on these kinds of social communications. Not something that you would normally think about, right? This kinds of vocal communication and so forth. You don't really think about that as being something that would alter gene expression in the brain. But I'll show you evidence that it clearly does. Now, I inserted the word neurogenetics up here just to make my own little piece about this. Again, as we've heard, you know, if everything were determined solely by our genes, we wouldn't have to worry about epigenetics because the genes would be read out and that would be the answer. But in fact, it's not. And so environmental conditions and so forth alter the books in the library that are read. And of course, but those processes actually occur for almost all the epigenetics we've heard about, briefly touched on by Francis, is talking about behavioral epigenetics, where the change that's occurring is in a specific tissue, is in the brain, and we'll hear some more about that, I think, from Michael Kabor later on. So I'm looking very specifically at what's happening in the genetic material in the brain in this kind of environmental experiential context. And the work started long ago in the distant mists of time, in the dark ages. It was like the 1980s. It was back before in the early days of DNA cloning and so forth. And before there were genomes, much less, nobody ever heard of all these acronyms and everything. It was so, but I got interested in these birds and so did some of the very first work. Well, did the first work on actually cloning genes from songbirds. And I'm I'm one of the ones that we came to, and I'm not going to give you the down and dirty to how we picked this gene and how we named it and so forth. But it is a gene that is still a major focus today. In fact, we heard Michael Meany talk about this. We heard Russ Fernald talk about this, and probably some other ones. It goes by different names. That's a little confusing. But in fact, you've heard it in the conference referred to as EGR1. You've heard it referred to as NGFIA. I'm going to make it interesting and call it by yet another name, which is really an acronym of all of these other ones. So this is a gene that has been linked to nerve growth factor, early growth response, these kinds of things. So this is one of the reasons why we started looking at this 25 years ago now. And one of my early colleagues and students, Claudio Mello, had this result that I have to show in every talk because it's still a really fascinating one and it's still really at the root of things. When we looked in the brain at the expression of this gene using an in situ hybridization technique where we label the probe and we're looking at a side view, a sagittal view of the brain, and you look up here and you don't see anything, and because this is not expressed at a very high level. But after a bird hears a song playback, 30 minutes in this case of a tape recorded song, you have this marked induction, this increase in this messenger RNA and the protein in this part of the brain that, as it turns out after about 10 more years of work in a bunch of different labs, is clearly the analog of auditory cortex and in a mammal. So this is a higher brain region, and this is not simply a sensory response. A key point here is this is not just a sensory response. There's more to it than this. So, for example, if when Claudio played the song of the same species, conspecific, and quantified this change, he got a high response. But if he played the song of a different species, he would still get a response relative to no sound, but it was half as much. And if he just played pure auditory tones, he got no response at all. So this is an auditory brain region that's responding to certain types of sensory, I guess sensory we'll call it. Certainly the sensory is the primary driver, but there's some kind of categorical process that's been applied to this. In more recent work, here's another illustration of that discrimination ability of this whole response. Yachi Lin has done an experiment where to get a very nicely, tightly controlled stimulus, so everything is the same in terms of the energy that's coming into the auditory forebrain and so forth, she created a stimulus we refer to as song envelope noise. That was the song. This is the song envelope noise. Okay, so we basically randomize the frequency content but have the same amplitude envelope, and that shows the same property of where this gene response is higher to the actual real song than to this enveloped noise analog of the song. But even that still actually gives a response relative to silence. So again, there's a very sophisticated some sort of processing going on having to do with perhaps the salience or the significance of this particular stimulus. Now, in the last 10 years or so, it has emerged that this particular gene is a profound player in epigenetic mechanisms. Early on it was identified as this so-called immediate early gene, this idea that it's a growth response, growth related. But it turns out that it actually mediates what can be called epigenetic effects in various ways. It encodes a transcription factor that binds to the promoters of many other genes. And this is a study actually done in microglia, a neuronal cell, a brain cell that's not a neuron, probably very important for remodeling and some of the other things that we'll hear about. And just a test showing that this particular EGR protein now interacts with all these different promoters. So this becomes a way for a change in one gene to have these ramifying effects through functional networks in gene expression. In the beautiful work of Moshe Ziff and Michael Beanie and so forth, of course, have also taken the same gene forward under the rubric NGFIA and shown how, as we've been hearing, how different kinds of maternal behavior, licking and grooming, can result in different degrees of methylation of DNA. And in fact, this same NGFIA seems to be part of the mechanism that directs changes in methylation to chromatin. So clearly I haven't actually thought so much about this in the context of epigenetics, but this is clearly an appropriate framework and an appropriate time to be thinking very, very seriously about that. So more time went by, okay? 20 years went by from the time we started studying all of this, and we went from having the first genes to having the genome. It was just published back a little over a year ago. Well, actually, it's almost 2 years now. But the whole sequence of the zebrafinch genome, and that's allowed us now to move into a different level of analysis and to ask questions about not just one gene, but to look much more broadly and systematically at what's happening in terms of gene expression in the brain and how natural experience is altering and modulating gene expression. And one of the ways that we're doing this now is with this technique of RNA-seq. I know Gene said the word. I'm not sure if anybody's really described it yet. I'm not going to go into any depth here. I'll just simply say that basically it's a sequencing-based method that instead of sequencing DNA, you sequence the RNA, the material that represents the output of the primary level of gene expression, and you sequence it, and from the sequencing you find out what it encodes, and from the number of times you count each sequence, that gives you a direct digital measure of how much expression is occurring. And so we've been able to do RNA-seq type experiments using the same brain region, this auditory forebrain, and map those reads back to the genome. And I'm going to come back to this slide in a minute. Just for the moment, I'll say that we can do this now and can do various kinds of quantitative analyses of gene expression using this approach. And so in one of our first experiments to really test this idea out, Sarah London and Chris Balakrishnan did the experiment I'm going to describe now. They have both, just in the last few months, moved on to independent positions. Sarah is now at University of Chicago, and Chris is at East Carolina. And so we still haven't published this. This will actually be part of our PNAS offering. So the experiment that they did was to ask the following question. Is there a difference in the profile of gene expression in animals, songbirds, that are hearing the song that's novel to them, they haven't heard this particular song before, compared to a song that they have heard before and has been made familiar, compared to not hearing any song at all? And the experimental design is shown here. All the birds were put into each individual bird was put into a sound chamber for a couple of days. And on this day here, all the birds heard about 3 hours of repeated playback of a song, the yellow song, if you will, song A. Then on the next day, birds in these three different groups heard either the same song, at that point it was a familiar song, or they heard a different song, or they heard nothing at all. And so in looking at that, we can make these different pairwise comparisons of the RNA that was present in the brain after this last day 2 playback experience. And we can basically do each one of these different sides. And I'm going to focus right now just on this comparison of what's different between a bird hearing an unfamiliar or novel song compared to a bird hearing a familiar song. Now, one of the really fascinating points, and something we had established in earlier work using microarrays, is that indeed these are both different from a bird that is not hearing anything. So just the sound of hearing a song playback results in very large, robust, complex changes in gene expression. Doesn't matter whether it's familiar or unfamiliar. But we see a very intriguing difference between the familiar and the novel stimulus in this case, suggesting again a kind of categorical process. There are about 31 genes that in this initial assay came up above the false discovery rate threshold, and they're just arrayed here in terms of the ratio. So a high number here means it's higher in the unfamiliar condition, the bird hearing the novel song, than it is in the bird hearing the familiar song. And so we have positive values here and negative values down here. And these are the genes. And so we can actually, in fact, typically what's happening here is that these are genes that are going one direction when a bird hears a familiar song and the opposite direction when a bird hears an unfamiliar song. When we look at what kinds of genes they encode, there's another really interesting piece to this, which is that there's a very strong functional clustering of activities here, with almost all of these genes that are increasing in the novel unfamiliar experience relative to the familiar one. They are heavily enriched for genes that have roles in cellular immunity, cellular defense processes. Not the kind of thing that you normally think about in the central nervous system, but perhaps part of this whole peripheral brain system. I don't know. It's something, a very interesting thing that we will be following up on. These things that are going down over here are more kind of traditional developmental type genes, retinoid signaling pathways and adhesion processes. So in my third little story now, my third little vignette, I want to come back to this mapping of gene expression in the songbird brain, where we found, as many other people have in other organisms, that a large proportion of the genetic output, of the books being pulled off the shelf, if you will, from in the library, do not actually encode proteins. Okay, the protein coding ones are this CDS, that's what that means, ensemble CDS. And you can see that 31% of these are intergenic. That means they're in parts of the genome that basically don't look like they code proteins, okay? And it turns out that in a lot of these, as well as some of these other genes, some of these other things down there in so-called intronic regions, actually are probably bona fide transcripts. They're genes that encode not proteins, but so-called non-coding RNAs. The RNA itself is functional. And this is a whole new category of gene expression that all of biology has been scrambling to try to make sense of in the last 5 or 10 years. And it's clearly really important in the brain. The subcategory of these non-coding RNAs that's been studied the most is the subcategory called the microRNAs, and we've heard a couple of, again, allusions to it, including Fraser's Last Dream or whatever. So I'll tell you briefly about that. MicroRNAs are simply short, highly conserved, and for the most part, sequences. They're 20 to 30 nucleotides, 20 to 22 nucleotides long. But there are about 1,000 of them, something like that, in different species. And their function has been most clearly shown in terms of targeting. Each one of these microRNAs targets a different set of messenger RNAs, and it will degrade them or suppress them. So this becomes a way to layer a new mechanism for modulating gene expression on top of the underlying transcriptional control systems. Now, it turns out that microRNAs are also emerging as being directly involved in some of those epigenetic processes that involve DNA methylation. There are at least 2 microRNAs that described in this review here where that act directly, again, on these enzymes and these machinery pathways. So this is clearly an important part of the epigenetic story. So for various reasons, we got very interested in this, and we did a direct sequencing analysis to test the hypothesis that hearing a song, would that actually alter microRNA expression in the zebrafinch auditory forebrain, and indeed it did. We found both known as well as a couple of novel microRNAs that had not been described in other species before that go up. For the most part, actually, they seem to decline very rapidly as soon as the song's playback starts. These microRNAs go down. I'll show you just one example of that. And that is one of the novel microRNAs. I showed you this same slide just a little bit earlier, where recall this is showing the zinc messenger RNA increasing whenever a bird is hearing a normal song relative to different controls. And these same animals exactly the same animals were analyzed now for the expression of this one particular microRNA. And you see here the opposite response in the same samples. It's going down in response to the song, and that response is quite specific. It's not changing significantly when the birds are hearing this noise enveloped stimulus. So there's discrimination of song quality at the level of microRNAs in the brain. Now, what does this all mean and what is it all doing? These are all questions that we have to figure out. But I think it's certainly, the brain is more plastic than we know. More is going on here than we've even begun to really think about. So to summarize then what I've told you about, I think we can say that experience, social experiences and so forth, naturalistic experience immediately alters brain gene expression on a time course of, as Russ so elegantly showed, minutes. And it does so by altering many different genes. It's not just a single gene. It's a complex response. And the response is surprisingly selective. It seems to depend on the quality and the nature of the experience. And finally, that this can lead to lasting long-term changes in gene expression. I didn't tell you a lot about that, focus on that here today, but certainly we have seen, again, evidence that early experiences can lead to long-term changes. So we really get a picture then of the epineurogenome, if you will, as a point of integration, a very deep integration. Integration across changing social conditions and perhaps integration across life experiences. Raising a bunch of interesting things to think about for the future. Does this epigenetic program limit or promote adaptive plasticity? That's a question that's at the root of a lot of the traditional biology and thinking about songbirds and their critical periods. It's obviously of interest, I think, to this audience. It's one of rather, I think we'd like to get beyond simply saying epigenetics happens, because clearly it does. But so what? What does it mean to have a change in chromatin methylation and in DNA modification mediated by microRNAs and whatnot? What does this do? We have the intuition that it probably is related somehow to adaptive plasticity. It's either good or bad. I don't know. So these are one of the things that I think we really want to be looking at in the near future. What are the long-term consequences of activating or suppressing cellular defense mechanisms in the brain? This is a very new finding, and I'm just kind of puzzling this one over. So if any of you guys have thoughts about that, I'll be very interested. And then finally, as Marla did touch on, how does individual genetic variation affect this epigenetic program? Because we are not all alike. We do have different genetic material, and it is going to interact with all of these epigenetic processes as well. And understanding the boundaries and the limits of that is I think, one of the big missions of this broad group and this broad community that we're engaged in. I will stop there and I'll take questions.